Good afternoon, respected faculties and everyone present over here. Myself, Dr. Ashwini Khankar. I'll be presenting a seminar on immunology of transplantation. My moderator is Dr. Yogesh Pavade sir, Assistant Professor, Department of Transfusion Medicine, MGM Medical College, Rainbow Hospital, Indore. The world's first successful organ transplantation was kidney transplantation, which was undertaken by David Hume and Joseph Kelly at Peter Brigham Hospital in Boston in 1954. The first kidney transplant in India was performed on 1st December at CMC Vellore. First successful transplants, 1954 first kidney transplant was done, 1968 first pancreas transplant, 1967 first liver transplant. These are the name of doctors and the universities where these transplants were taken place. First living liver transplant was taken place in 1989 and first living lung in 1990. An organ transplant is a surgical procedure in which a failing organ is replaced by a functioning one from a donor with a compatible tissue type. The transplants can be classified into the grafts and can be classified into autograft, allograft, isograft, xenograft, orthoptic and heterotopic graft. Autograft is when a person takes a graft from himself, the same body. Isograft is when a person takes graft from his identical twin. The sources can be living donor or cells for autograft, cadaver on animals. Autologous can be examples skin or artery transplants, and these are generally not rejected for the same individual is the donor and the recipient. Isograft can be any organ among identical twins, and these are also generally not rejected. Allograft is within the same species when human A and human B are the donor and recipient respectively, and the grafts can be kidney, lung, and heart transplants. Xenografts are interspecies grafts which are generally rejected unless non-antigenic. Now, transplant immunology. It is the rejection caused by genetic differences between the donor and the recipient, triggering immune responses. The antigens are HLA and ABO antigen mainly. The allo antigens are the antigens which vary between members of the same species, and the reaction to these antigens is called allo reaction. And in transplantation, these reactions can be either host versus graft or graft versus host. The first antigen, HLA antigen, it is also called the MHC complex, major histocompatibility complex. Its gene location is short arm of chromosome 6 and it contains about 224 functional genes. It is expressed on the surface of human cells and it encodes proteins which are responsible for the immune system. The differences between the HLA molecules are responsible for the immune responses which are provoked by T cells, B cells and NK cells, natural killer cells. MHC has two classes, class 1 and class 2. The difference is as follows. The structure for class 1 has alpha and beta 2 microglobulin chain and class 2 has alpha and beta chain. The antigen source for class 1 is intracellular pathogens while for class 2 are the extracellular pathogens. The T cell interaction, the immune response responsible for class 1 is CD8 T cells, cytotoxic T cells, and for class 2 are the CD4 helper T cells. And immune function by which they cause the immune responses, class 1 causes by cytotoxicity, in class 2 by the regulation and coordination of the other immune cells. The location for class 1 is on all nucleated cells and for class 2 it is primarily on the APCs, antigen presenting cells. This is the diagram. Class 1 molecule has, as you can see, alpha 1, alpha 2, alpha 3 linkage and beta 2 microglobulin, and class 2 has alpha and beta linkage. The inheritance of the MHC or HLA haplotypes are in Mendelian fashion. HLA on each parental chromosome is called HLA haplotype. These are codominantly expressed and each parent provides one haplotype to the offspring. Statistically, 25% siblings will have 100% identical match, 50% will have chance of only one haplotype, and 25% will be there who will have zero haplotype matching. HLA cross matching is done before transplanting any organ. It is three types: CDC cross match, complement dependent cytotoxicity cross match, flow cross match, and solid phase cross match. CDC cross match contains complements. It activates complements and the The donor's lymphocytes, recipient serums are added, complement is added to them, and the result is read by flow under microscope. In flow cross match, anti-human immunoglobulins are added to look for the compatibility, and in solid phase cross match, Luminex beads are added, and then those are read. Next, the ABO incompatibility. It is of less importance than HLA compatibility. ABO incompatibility can result in hyperacute rejections, mainly in vascularized grafts, such as those of kidneys and heart. Now, the role of transfusion services as our department includes immunohematological workup, donor assessment, follow-up, then transfusion support before, during, and after the surgery, and desensitization. The immunohematological workup consists of initiated well notice, a prior notice should be there of the surgery. We should have a sample from both the donor and the recipient, and we are supposed to perform the following tests: ABO and RHD grouping, titers of anti-A, anti-B grouping in case of mismatching, direct antiglobulin test, and alloantibodies in case they are detected and then identified. Therapeutic plasma exchange also plays a role in solid organ transplantation by it reduces the risk of rejection as it can remove donor-specific antibodies. It can desensitize patients before the transfusion. It can treat antibody-mediated rejection. It can also be useful for treating transplant-associated thrombotic microangiopathy. It also is useful in remission of autoimmune diseases when combined with immunosuppressive agents. During TP, as we know, patient's blood is removed and passed to a device that separates the plasma from the